What'd you say about me, Joe?
I don't. Oh my god. Alright, so to start this project, I'm gonna put some music on to yeah, just to yeah. So it's a good song too though. Okay, so starting off with the interface, which is the longest and the most highly active preparatory stage of the cell, well the cycle, where the eukaryotic cell, it grows and it replicates its DNA and it prepares for a division, which then goes for the prophase, which the prophase is the first stage of mitosis, well the phase, and it is the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Then that's where we hop into the metaphase, which is the second stage of cell division between prophase and anaphase, during which the chromosomes become attached to the spindle fibers. And then after the metaphase comes the anaphase, which is the third stage of mitosis and meiosis, where sister chromosomes, or homologous chromosomes in meiosis one, separate and are pulled toward the opposite poles of the cell, ensuring each daughter cell receives an identical set of chromosomes. And then we finally got the telophase, which is the final stage of mitosis, or meiosis, where separated chromosomes arrive at opposite poles, which is like the nuclear envelopes reform around each set of daughter chromosomes, and the mitotic spindle disassembles. Yeah. Then we could also squeeze in cytokinesis, which is the physical process of cell division. Right after that comes mitosis, which is the process of nuclear division in eukaryotic cells that produce two genetically identical daughter cells from a single parent cell. Which is, I'm pretty sure that's where all the other things have to like... Oh my gosh, my bad, my dog is... And sister chromatids are next, which is two identical copies of a single replicated chromosome, which is like basically a copy, hold on, oh my gosh, I already said that. It's like a clone, which is formed during the S phase of the cell cycle and is joined at the centromere. And then a centromere is a specialized, highly repetitive DNA, which is a DNA region on a chromosome that links sister chromatids, which we just talked about, and serves as the assembly site for the core. I don't know how to say that. And the X's, which are the chromosomes, which are thread-like, highly condensed structures of DNA and histone proteins located within the nucleus, acting as the primary storage for genetic information. I need some water, hold on. Then right after that is the DNA replication, which is a semi-conservative, high-fidelity process of copying cells' DNA before division, which is the S phase, producing two identical double helices, which each with one new and one original strand. Then after that would be the chromatin, which is a complex of DNA, and RNA, and protein, primarily histones, located within the eukaryotic nucleus. Then the spindle fibers, which are microtubule protein filaments that form in the mitotic spindle apparatus. Whoa. And centrioles are cylindrical paired organelles found in animal cell chromosomes, composed of 9 triplet of tubulin microtubules arranged in a ring. And Then next comes the nuclear envelope, which is a highly regulated double membrane barrier in eukaryotic cells that separate the nucleus from the cytoplasm, protecting the genome. And then the cytoplasm is a gel-like substance composed of water, salt, and organic molecules that fills the interior of a cell and house organelles. And then the organelles are specialized membrane-bound structures within eukaryotic cells that perform specific functions similar to organs in the body. Then daughter cells are new cells produced through a division of a single parent or mother cell during mitosis or meiosis. Nuclei are fundamental dense centers of systems existing as cellular organelles in biology or atomic cores in physics. And last is the metaphase plate, which is an imaginary plane equidistant from the two spindle poles where chromosomes align during the metaphase stage of mitosis and meiosis.